All right, this is our first video in our lecture series on development. And I think you'll notice as we kind of try to speed through an entire human life, basically, in how we cover different things in this unit, it's especially clear here that this class, can only really touch on a lot of different surface ideas. So just a reminder that as you're finding things that interest you across these different units, keep notes about that. Keep that in the back of your mind for when you're perhaps registering for future psychology classes, and maybe you want to, take developmental classes or whatever might catch your fancy in other units. But in this developmental unit, we're going to go pretty quickly through a few different things, how people develop physically, and then more psych-focused topics, how people develop intellectually and socially and personally. But I think you'll also see that the physical development side of that really informs a lot of that intellectual, social, and personal development. Because as we've talked about, the physical processes that are underlying our behavior and the way we perceive and think about the world are really inextricable. The physical processes are inherent to those psychological processes. So you'll see those connections there. The way I want to start, though, is just to briefly mention that we've talked about experiments as being very important tools for psychologists so far in this class. That's partly from my training as a social psychologist. That's our preferred method. But it's not the only method in psychology, obviously. When we get further on in the semester, talking about disorders and treatment, we'll focus a bit more on case studies as well, so you'll get a feel for some other methods there. But in developmental psychology, there are a couple of different tools that both have their pros and cons. And you need to have this toolkit because experiments are really, really difficult to conduct in developmental studies. Imagine trying to see how someone develops based on their random assignment to some condition. How can you keep that condition constant in someone's life? How can you make a decision about something they'll be exposed to for a long enough time to see if they develop differently? Well, the answer to that most of the time is that you can't. So the two main ways that developmental researchers are going to look at how people change over time are through cross-sectional and longitudinal designs. And these words may sound more or less familiar from other classes or other things you've seen, but just to give you a quick rundown, cross-sectional developmental designs are when you randomly assign, ideally, or sorry, randomly select, ideally, people from different age groups. You're taking different cohorts or segments of the population and looking at, perhaps people that are 2 years old and 5 years old and 8 years old and 11 years old, 14, and on and on and on to see the differences in their abilities and their development. So being able to do this is an attractive way to study something because you can do it all at once. You don't have to wait for someone to grow up. You can collect data and compare 2-year-olds to 8-year-olds and 5-year-olds to 14-year-olds and see what those differences are. So it's easier to get this kind of data. It's usually going to be cheaper because you don't have to administer it over time, and it's going to be relatively fast because you could just collect all the same data from those different cohorts or segments of the age groups and compare it immediately. But when you are comparing these different groups of people that already are pre-existing, it's really difficult to rule out alternative explanations. So one useful example might be that we all grow up in different historical contexts, right? The way that a natural disaster, for example, might affect people that are children is going to be different than how it's going to affect people that are adults. So thinking about things like Hurricane Katrina in New Orleans, even winter storms recently that have been causing problems across the country. That might affect different people in different ways, and you can't really account for someone's entire life history when you are trying to figure out what might have influenced their development. So it's really difficult to pull that apart. You also have a really hard time drawing any cause and effect conclusions because, as we know, it doesn't conform to experimental design sorts of standards. But also, there are some more sophisticated statistical methods that you can use to look at causality if you have data measured at a bunch of different times for the same group of people. And cross-sectional data is measuring once for a bunch of different groups of people. So it's a little bit different. So these different unique experiences that different cohorts of people are going to have might have problems, and the fact that you can't really make causal claims is another big issue. The alternative to cross-sectional designs, or an alternative, would be longitudinal designs, where you collect data from a single group of people, usually recruited when they're relatively young, maybe even in infancy, and follow them over time, seeing their development on different things about their cognitive abilities or their morals or their personal development in some way. So if you do this, you can make a much stronger argument about aging being the causal factor influencing someone's development, where that might be a little bit more difficult in a cross-sectional design. So if you're drawing conclusions about your specific generation of people that you're studying, cohort effects might not be a problem. But if you only study, like let's say, for example, that your study has only looked at boomers, people that were born relatively soon after World War II, usually like 1945 to 1965 or something like that. The conclusions that you draw about baby boomers might not apply to Gen Xers or millennials or Gen Z people, right? There might be something about your conclusions that's tied to that group of people. So you might be right in saying something about the developmental trajectory of those people, but it might be wrong about earlier or subsequent generations. So that's a big disadvantage, that your conclusions might not generalize past your sample or your generation you have looked at. It also takes a long time. If you want to make conclusions about somebody's entire life history, they have to live their lives, right? So there are longitudinal studies that have been going on at this point for 30, 40, 50 years, trying to look at people's development. So that requires a lot of buy-in from research teams and being able to groom and train, new generations of researchers and keep things like that going. So it's kind of a somewhat risky way to do it, and it might not ever have benefits for the people that start it, basically. You also are going to see things like some of your participants are going to die or not be able to be contacted anymore, and that might lead to problems in how you're making conclusions about your results. You might see people are just getting good at the same tests if you're giving them to them over and over again. And participants might also think back to things they've said in these studies or done and try to be consistent over time, which can kind of undermine your ability to see changes. So maybe a child is doing like some sort of conservation of volume test that we've seen before, and they know the right answer, but they remember what they've said before, so they just say that same thing. That would undermine your ability to say anything conclusive about that. And finally, reactivity. People being studied are just liable to act differently than people in their natural environment, right? So that type of reactivity to the types of measurements that you're taking might also be a problem. Finally, another way that developmental researchers look at this stuff is actually to combine these things to help offset some of these problems that one or the other method might have. And we call this a sequential method. And what a sequential method is taking cross-sectional measurements, but following those cross-sectional cohort groups across time. So your example might be you have people in your study that were born in 1970, some in 1980, some in 1990, some in 2000. So you have these cross-sectional cohorts, but you still follow them over time. So you measure in 95, 2005, 2015, 2025, and you take this over time to be able to both see the development within a cohort and be able to compare between cohorts and see these differences compare to one another. So this can be a really, really useful thing to try to offset some of those problems that each of these designs have. However, it is enormously expensive and administratively burdensome to try to do something like this, right? I already told you longitudinal designs are problematic. Well, imagine doing 4 separate longitudinal designs at once, right? So these are ones that people are only going to be able to do with a lot of support from things like grant money and a lot of administrative buy-in. So now that we have a little bit of an idea about how people study developmental changes in psychology very broadly, let's talk a little bit about the physical development that happens in a person. And a lot of this development, of course, is going to start before they are actually born. So people obviously start out when a sperm cell fertilizes an egg cell. They become a single cell called the zygote. And within 9 months, that single cell has multiplied into literally billions of cells that compose the entire newborn. After about 2 weeks of that first cell coming to exist, that cell implants in the uterine wall. It starts to form the different support systems around it that are going to keep it alive or keep it developing over the course of the pregnancy. And only about half of the zygotes that are fertilized actually make it to this point. So there are a lot of fertilized eggs that are actually not being implanted and having the trajectory to become a fetus and eventually a newborn. But over that next 6 weeks, that little clump of cells, little ball of cells, is going to start looking like something that you might recognize as kind of mammalian into an embryo, this embryonic period. And if that embryo has a Y chromosome, so male human beings have an X and a Y chromosome, female human beings have two X chromosomes. And there, of course, are different biological sexes too that have different combinations or have themselves express themselves differently. But those are the most common two. But if there is a Y chromosome, testosterone production and secretion will start to create some of those primary sex characteristics. Then about week 9 is when we would start calling this organism a fetus. There start to be different solid structures developing, bones and muscles. They start to move when people are getting what's the word? I can't remember. The ultrasounds or sonograms. They start to see the fetus moving around while they're doing that. And in the last 3 months of the entire pregnancy period, there is an explosion of growth as well, where the body and the brain become much, much more complex. Different systems finishing up their production and the brain growing in size quite a bit. And I think this is very well known now, but not too long ago, people didn't really understand that the environment that the mother is herself in and the different things that she is allowing into her body are enormously influential in the development of the child, the fetus. So anything that is influencing the fetus in a negative way is generally referred to as a teratogen. These are things like viruses, bacteria, cold, any other problems that might happen medically, and drugs like alcohol, nicotine, other things as well. And when the fetus is most susceptible to these different types of teratogens for different systems to develop are called critical periods. So for different types of structures in the body, there are different critical periods. And if you look at this developmental chart here, going from pre-embryonic in the first couple weeks all the way up to the end of the embryonic stage at about 8 or 9 weeks, and then this kind of compressed fetal period here, you can see that the early development, just in the first several weeks, is enormously important for a lot of these different systems. The heart, upper limbs, lower limbs, they can be disrupted by certain illnesses and certainly by drugs and alcohol. And some of the more important structures or more far-reaching structures, let's say, like the central nervous system, they have a longer critical period here in orange. These purple periods are going to show times when those systems still might be damaged by teratogens, but maybe not as much as in these critical periods. So critical periods, again, are the ones where they're most susceptible to these types of influences. Although, it's never like helpful to have alcohol introduced to a newborn or a fetus's system, clearly. But they do the most damage in development, this really early portion for most of these things. In the next video, I'm going to move on to actual birth. Well, not that much detail about birth, but actually talk about what happens after a child is born.